Hi everyone, I'm Manuel Urbaz Oliveira and today I'm going to present you my seminar that is about an investigation of a nosocomial outbreak of fungemia caused by Candida pelliculosa in a Korean tertiary care center. So we have the Candida pelliculosa is a rare pathogen causing invasive infection. It's a type of unicellular fungi also called yeast and it can be usually found in fruit, soil and plants. It is also known as Hansenula anomala or Pichia anomala. The first infection with Candida pelliculosa was reported in an infant who died of interstitial pneumonia in 1953. Infections caused by Candida pelliculosa are characteristic in children and in severely immunocompromised patients. What is fungemia? Fungemia is the presence of fungi or yeast in the blood. Some risk factors for developing fungal infection include the use and abuse of central venous catheters. Symptoms can range from mild to extreme, and they are often described as flu-like symptoms. Many symptoms may be associated with fungemia, including pain, chronic fatigue, acute confusion, and infections. What is a nosocomial outbreak? A nosocomial outbreak is a significant increase of the infection rate taking place in a hospital or acquired in a hospital. Also, in the study, they describe an epidemiologic outbreak investigation of 11 cases of fungemia caused by Candida pelliculosa during 24 days in a single center in Ulsan, South Korea. Objective: we have that this study followed up on a fungemia caused by Candida pelliculosa in a medical center, and they aim to find risk factors of the spread of this pathogen by analyzing each case thoroughly. Material and methods. The antifungal susceptibilities were determined using BTEC2. BTEC2 is a system that uses cards with colorimetric regions, which are inoculated with a suspension of a pure microbial culture and the development profile is interpreted automatically. There are four types of reactive cards available for the identification of different classes of organisms. We have GN, that is for gram-negative bacilli fermenters and non-fermenters. We have GP, that is for coccus and bacilli that do not form gram-positive spores. We have YST, that is for yeasts and yeast organisms. And we have BCL, that is for gram-positive spore-forming bacilli. Material and methods. We have two processes that are important in the article, that are the study design and the DNA extraction. For the study design, they describe clinical characteristics of patients with Candida pelliculosa fungemia. And it is important, in this image we can see that 3 of the patients had multiple trauma. So they were a lot of time in the hospital and they had a lot of time as central venous catheters. And 2 of the patients died because of the candidemia. And the DNA extraction is really important because of the processes that they realize to the amplification of the DNA. We have the randomly amplified polymorphic DNA assay or RAPD assay. It is an enzymatic amplification of polymorphic fragments of samples that behave randomly when expressing the result. It is a type of PCR, and the DNA fragments obtained by means of this technique are amplified in random regions of the genome, since the primers of the reaction are arbitrary sequences of synthetic DNA. This technique is based on the use of a single 10 base pairs oligonucleotide that is random, that hybridize randomly with the DNA under study. The polymorphisms observed between different individuals consist of the presence or absence of amplified DNA fragments. The information it gives us is the presence or absence of bands. So we have here an image that summarizes the process. That first of all is the DNA extraction. We have the sample A and sample B that are the ones that we use here, and the amplification that is with DNA polymerase and short universal primers. We have the hybridization and the electrophoresis to look for the results. We have also the matrix-assisted laser desorption ionization, or MALDI-TOF. It is an ionization technique that uses a laser energy absorbing matrix to create ions from large molecules with minimal fragmentations. It is based, first of all, for the sample. It is mixed with a suitable matrix material and applied to a metal plate. Then a pulse laser irradiates the sample. It triggers the ablation and desorption of the sample and matrix material. The analyte molecules are ionized by being protonated or deprotonated in the hot plunge of ablated gases, and then they can be accelerated into whichever mass spectrometer is used to analyze them. Also, we have here an image that summarizes the process. We have first of all the acceleration, we have the separation, that is the top part, we have the detection, and the result, that is for the spectrum. Results. So we have the results for the antifungal susceptibility testing, that is based of 30 Candida pelliculosa isolates from the 11 patients. So it is important in this image that 2 of the antifungal agents had a minimal inhibitory concentration. There are 2 isolates with 0.5 mg liter of amphotericin B and 2 isolates with less than 1 mg liter in the fluconazole. So the rest were susceptible to amphotericin B, fluconazole, voriconazole, caspofungin, and micafungin. It is important that caspofungin and micafungin are also known as echinocandin. We have in this figure the results of the RAPD assay. In the agarose gel electrophoresis of polymerase chain reactions, that is a product of 4 Candida isolates with a specific primer of Candida pelliculosa. It is important that in this number 10 we have this band that is a little clearer than the rest. And this is the size marker that says the base pairs to compare each one. And at the end, we determined that these 4 outbreak isolates were identical in the APD assay. We have also the epidemiologic plot of Candida pelliculosa fungemia, and it says the day that they found out the detection of the outbreak, that was November 30 of 2015, and the different places that of this nosocomial outbreak, that was in Ward 1, was in trauma ICU, medical ICU, and Ward 2. Also, the intervention for infection control. So they began the intervention including hand hygiene education, isolation of all patients with Candida pelliculosa fungemia in a single room, aprons and gloves were worn for contact. Also, they had a suspicion that the use of identical X-ray machine as a probable source of transmission of Candida pelliculosa. So, they disinfected the X-ray cassettes using alcohol and quaternary ammonium compounds to see if it was true or not. Also, here we have in the discussion 3 groups of authors that analyzed some risk factors, and all of them were right and they were used in the article. Conclusions. It is important in medicine to know the risk factors of nosocomial infections so that they can be prevented and treated in case there is not a good management of the implements within the intensive care units or any space around a critical patient, because we know that these patients are also severely immunocompromised, that is a risk factor for the candidemia or any other infection. Also, having knowledge of these pathogens that can cause fungemia or any other infections is the key of a good care in every medical center, because it makes us aware that it can happen at any moment, and if we suspect of a specific pathogen or bacteria, we can diagnose quickly and start the treatment preventing an outbreak. In this case was with candidemia in Candida pelliculosa. So thank you for your time, and this is my mind map.